We give it 1 minute and we can start.
Yeah, please feel free to start.
Okay, hello and welcome to the Institute for Molecular Science and Engineering's webinar entitled Science and Engineering Innovations as part of interdisciplinary approaches to preventing neglected tropical diseases. And this will be presented by Professor Michael Templeton from the Department of Civil and Environmental Engineering here at Imperial. The webinar is part of the Water, Sanitation, Hygiene for Preventing Infectious Diseases webinar series. I'm Professor Nick Vulvoulis, a professor of environmental technology at the Center for Environmental Policy. We will be putting some helpful links in the chat, including links to the Institute's Eventbrite page, when you can find the registration for all our upcoming webinars, the YouTube channel where all our previous webinars are uploaded, and also links to the blog, the Never Lick the Spoon podcast, and our previous briefing papers. You can also register for our newsletter so that you keep up to date with all of the news and events from the Institute. Just a few top tips to make sure that we have a great webinar. Please make sure that your microphone and video are switched off. If you wish to ask a question, please put your question in the QA function at any time during the webinar. Your questions will be answered at the end. Please do not put questions in the chat function. If you have any technical difficulties, please message IMSE Imperial privately, and a member of the team will try and help where possible. I will now hand over to our presenter and introduce him first. Michael Templeton is a professor of public health engineering in the Department of Civil and Environmental Engineering at Imperial College London. His research focuses on addressing public health challenges related to water supply and sanitation. He is a fellow of the Institution of Civil Engineers and has served on strategic advisory groups and technical review panels for the World Health Organization, the UK Research and Innovation Global Challenges Research Fund, the Natural Sciences and Engineering Research Council of Canada, and the Swedish Research Council. He is the co-chair of Imperial's Global Development Hub. Michael, the floor is yours.
Thank you, Nick, and thank you to the IMSE webinar series organizers for inviting me to speak. So today's talk, so as Nick said, I'm a public health engineer, so I'm not a molecular scientist, but there will be a little bit of molecular science in the lecture today, I think. But really, I think the key sort of theme or message that I'd like to get across from the presentation is about the fact that scientists and engineers have an important role to play in addressing a lot of global health challenges, including what I'm going to be focusing on today, which is neglected tropical diseases. But that really, the impact of science and engineering is only really realized fully or reaches its full potential if it's part of interdisciplinary collaborations. So even if you're not interested in neglected tropical diseases, maybe the model that I'll be presenting today of how I've gone about some research on this topic will be of interest to you and something that you might want to think to replicate. Because I think interdisciplinary collaborations, you know, they rely on people being really good at their own disciplines, obviously, but also being willing to work with people with very different perspectives, but who have something else to contribute to solving the problem. And I think, you know, I think we've all become sort of familiar with this idea that a lot of health challenges require an interdisciplinary kind of approach since COVID, you know, came about. Where we had people developing vaccines, but we also had people thinking about ventilation and about masks and, you know, how we move people safely around urban environments. And so it took really people working on the problem from different angles to protect people, try to prevent infection, and then also to treat people once they became infected. And sadly, a lot of neglected tropical diseases around the world, there are no vaccines. So really, we need, you know, the importance of preventing infection is even higher for some of these diseases. And sadly, a lot of people in low-income countries aren't able to do so currently. So I'll be approaching this really from the standpoint of a civil engineer who's interested in water and sanitation, but I'll be showing how other disciplines have contributed to this research also. Right. So you might be wondering, what is a neglected tropical disease? So it's called NTDs for short. And it's actually a series of diseases that have been added to a list by the World Health Organization. And really, these things have a few things in common, these diseases. One is that they tend to mostly affect the poorest people in society. And it's estimated that about 1 in 7 people globally have a neglected tropical disease. Sometimes people say that neglected tropical diseases are also about neglected people, really, because it's people who live in conditions which, you know, are always exposing them to the potential to contract these diseases. Many NTDs also are neglected because they're quite debilitating, and there's some often stigma associated with them, which means that they tend to be issues that sometimes countries want to sweep under the carpet and not really put a lot of focus into addressing. And also, as I said, they're diseases of poverty. So really, from an economic standpoint, there's been a lack of a driver historically for a market to pay for drug development for some of these diseases. And luckily now we've got to the stage where for a lot of these illnesses, the pharmaceutical companies actually do have effective treatments and actually donate them on very large scales. But as I said, we don't have vaccines for all of these diseases, and so reinfection is quite common if people continue to be exposed to the vector. So today I'm going to be focusing on one particular NTD, which is called schistosomiasis, which I might say schisto for short occasionally, because schistosomiasis is a bit of a mouthful. And it's also known as bilharzia, which you might have heard of before. And so schistosomiasis is a disease that's endemic in the global south. Most cases of schistosomiasis are in sub-Saharan Africa. It's estimated that about 200 million people are affected by schistosomiasis. So either are infected or are at quite a high risk of getting it at some point. You can see that it's also in pockets of Asia and also in South America. And there are a few different types of schistosomiasis. The sort of two most prevalent ones in Africa being what's called Schistosoma mansoni and Schistosoma hematobium. And so what the disease entails is the transmission of what are called schistosomes. So that you get the disease by having an adult worm, which is living in your body, and it tends to live in blood vessels around the intestine or the bladder. And you get these worms into your body by being exposed to contaminated water through water contact. So from your skin being in contact with water that contains one of the stages of the life cycle of these worms. And then once the worms grow in your body, they then release eggs, which leave your body in urine or feces. And one thing to note particularly about this disease is that there's what's called an intermediate host. So people who are infected, they shed the eggs into water if they don't have access to sanitation, to toilets. And then people are exposed by being exposed to the form of the parasite that comes out of the snails, which acts as an intermediate host in the environment. So these snails are particular types of freshwater snails, and some images here of the different types of snails depending on the form of schistosomiasis you're talking about. And it does take some expert knowledge really to know what type of snail to look for. It's not just sort of any old type of snail that can be the host. And some pictures here of what one of those intermediate hosts looks like in terms of how big the snail is. And then some zoomed in images here of an infected snail that's shedding what are called schistosomes cercariae. And this is the form of the parasite which swims around in water and can penetrate into human skin and start the whole infection loop again. So the little white lines that you see there in the middle image with the arrows next to them are blown up there, and that's the cercariae you can see on the right there. So in international development, people sometimes talk about what are called wicked problems. So these are problems that have been around for many, many years, and they're called wicked because they're very difficult to solve, because it's a very complex, there's a range of complex causes for these problems. And schistosomiasis definitely, I would say, falls into one of those categories. And this diagram sort of will help to illustrate that, I think, where this is a diagram from the Centers for Disease Control. And you can see here the life cycle I just talked about, where there's a person who is infected. They release a form of the parasite into water, which infects a snail, which is number 3 on the diagram. And then the snail eventually releases the cercariae, number 5 in the diagram, back into water. And it's those that penetrate into human skin. And if you look at the mathematics of it, it takes actually only a very small number of eggs reaching the water for large numbers of cercariae to be produced. So the snail essentially acts like a multiplier of the problem. This is quite important, because what it means is that unless you get very high levels of prevention of pollution of water, you're going to continue to have a very high risk of exposure and getting the disease. So it's a very effective disease pattern that has been established here. And as I say, it's a disease that's millennia old. People have even found evidence that perhaps the ancient Egyptians even suffered from schistosomiasis from contact with contaminated water in the Nile. And so if we look at the life cycle again, and we think about how could we cut this life cycle and solve this wicked problem, there's a few things that we could do. We could give drugs to people to kill the adult schistosomes in people's bodies if they're infected. And there are effective deworming drugs, which, as I said in the introduction, are now available as donations from the pharmaceutical companies. And they're very effective. The problem is that you're looking at hundreds of millions of people, so very, very large populations that you have to distribute the drug to. And also the fact that it's not a vaccine. So again, people, if they might take the drug, have the infection cleared, but if they continue to be exposed to contaminated water, they're going to get reinfected. Another thing we could do is we could install a lot of toilets, so that people, even if people are infected, there's no route for the eggs to get back into water and infect the snails and keep the whole cycle going. But again, as I showed earlier, probably you'd need very high coverage of sanitation for you to completely be able to break the cycle. Another approach that's been suggested and tried in a few places is maybe you could just kill all the snails that are the intermediate host. Although there are some concerns there potentially about the ecological effects of dosing molluscicides into lakes and rivers to kill snails. And probably, again, you'd have to get a pretty high coverage level of killing of the snails to completely cut transmission. And then a final thing you could do is if you didn't have the toilets, you weren't killing the snails, you could just assume the water is contaminated and develop ways to make the water safe. So to take the water out of the lake or the river, treat it, and make it safe for people to use for their water contact activities. So really, you can see here there's a few different barriers to protecting people from schistosomiasis. And historically, there's been maybe research in sort of siloed areas into each of these potential interventions. And increasingly, the World Health Organization is saying that you need to really have combinations of these approaches in order to try to approach elimination of schistosomiasis. And so, as I said earlier, you sort of have to think a bit of the math of the life cycle. And so if you look at sort of the diagram on the left here, where if you had a community with sanitation and one on the right without sanitation, in terms of having access to toilets, you can see on the left here, some of the Miracidia are probably going to end up in the toilet. But if some of them, even a small number of them still go into the water, maybe because not 100% of people have access to toilets, or there's occasional lapses in use of the toilet, then really, you're still going to have quite a large number of cercariae being produced and the infection cycle continuing. Compared to the situation on the right here, you don't really see that much of a difference. So probably in terms of sanitation, while it's definitely a very important intervention, and obviously having a toilet has lots of other benefits besides schistosomiasis prevention. Probably you're going to need very high community coverage levels before you're likely to have an effect. So what we did in our research is we decided we would focus on the last intervention in particular that I talked about, which was about assuming that the water is contaminated and that people need to, tomorrow they need to continue to have contact with water for various activities like doing laundry or bathing, or even for collecting for drinking water. Maybe we should look at how can we make the water safe for those activities. And so really, while there's a bit of a complex link between sanitation and infection, it's probably more logical or easier to show that the water contact is more directly related to the rate of infection. And as I said, to be effective, you need to think about providing safe water to people, not just for drinking water, which tends to be what a lot of NGOs focus on, giving people taps for drinking water, but also thinking about all of the other reasons that people have water contact and how can you try to accommodate that. So for example, doing laundry or bathing. And also another complex part of this is even if you did provide safe water to people, you need to understand why people might still prefer to go to a contaminated water source. So we need to also understand why do people have water contact, and are there sort of social or cultural practices or traditions around contact with a particular water source. I'm going to tell you a bit about a research project that was funded through the Global Challenges Research Fund. But before I do so, to summarize some work that we did before that. And the first thing we did is we did what's called a systematic review and meta-analysis, which those of you who might work in the medical field, it's something that's quite commonly done in medicine, where you look at all of the studies that have been published on a particular topic, linking two factors or linking factors to a certain health outcome, and you combine the data from all those studies in what's called a meta-analysis. And by combining the data sets, you can come up with very strong, higher orders of power conclusions. And so we did that in terms of looking at all of the studies that have been done in terms of access to water and sanitation and infection with schistosomiasis. And we found there that actually there were a lot of limitations of previous studies. They didn't always describe, for example, what type of sanitation people had in a community, or what the level of coverage of sanitation was in terms of the percentage of people having access. And also quite a few studies had confounding factors, like it was comparing people in very wealthy communities to people in very poor communities, where there could be a range of different reasons why people have more or less contact with contaminated water, besides just having access to a toilet, for example. And so what we did after that was we were part of a study with a lot of collaborators in Ethiopia, where we were really lucky and able to go around to about 1,600 schools in Ethiopia, where health data was being collected on about 50 children per school. So it was about 80,000 kids in total. And it was looking at measuring things like infection with schistosomiasis, but also other things like anemia and stunting and hookworm infections. At the same time, what we did is we went into the schools of those kids and we tried to be very descriptive and quantitative about the quality of the water and the sanitation that they had in those schools. And what we found basically was that actually the quality of the sanitation or the access to sanitation didn't have a statistical effect on the risk of these kids getting infection with schistosomiasis. It did with some of the other worms we were studying, but not so much with schistosomiasis. Whereas the kids who had to go to collect water several times a day because they didn't have a piped water supply in their school, they had a significant, statistically significant link with schistosomiasis. So that was interesting and sort of, again, pointed us towards maybe we want to focus on the water side of things in terms of protecting people right away from schistosomiasis. So we then started this project called WISER for short, which stands for Water Infrastructure for Schistosomiasis Endemic Regions. It's always good to have a snappy acronym to get Research Council funding. And our webpage there, wiserschisto.com, is still active if you want to go on there and read about the outputs. And there's also a blog and some news items that are updated on there. And so WISER was a 4-year project, which was funded through the GCRF, the Global Challenges Research Fund. And it was a really exciting project for me because I was the PI, but it involved a very interdisciplinary and international group. So there were water engineers, but there were also parasitologists, snail experts, synthetic biologists, anthropologists, and even some actors. And I'll tell you later on what the role of the actors was in the research. And it was also an international partnership. So researchers at Imperial and then the Natural History Museum in London teamed up with the National Institute for Medical Research in Tanzania and also Addis Ababa University in Ethiopia, which are 2 countries in Africa where schistosomiasis is a big problem in certain regions of those countries. So it was fantastic to work with people in different disciplines who can bring knowledge of different parts of the transmission cycle and different elements of the overall solution to eliminating the disease. And also to be able to work with international partners who can provide the local context, the local understanding of why disease transmission happens in certain settings, and really to also contribute intellectually in terms of what would be practical as solutions for these particular settings in these countries. So hugely valuable to have them as collaborators on the project. And so the specific objectives, the first one was sort of an engineering and science kind of objective, which is really if we were to treat the water, what kind of chlorine doses would we need, or how would we design sand filters, or could we use even UV or solar disinfection to make the water safe? So that's really sort of right in my wheelhouse, or my research group's wheelhouse in engineering, because it's sort of taking established water treatment methods, which are used already in low income settings, and testing whether they're good against this particular parasite. Second objective was more of a synthetic biology objective, which was looking at, can we develop better ways or sensors for detecting the cercariae in water bodies and assessing the contamination of water bodies. A lot of the methods that are used nowadays, they rely on people, as I said earlier, going out and finding those snails and then taking the snails back to the lab and seeing if the snails are shedding the parasite. And it takes a lot of expertise and time. And what we ideally wanted to do was to see if we could develop a rapid sort of in-field test method where you could take a water sample and somehow it would tell you quickly, what the risk level was of that water body in terms of the cercariae content. And finally was a more social science objective, which is about, let's say we can come up with engineering solutions and we can assess the risk of contamination of water bodies. We also need to understand, how do we sustainably implement these solutions, and are there sort of health awareness challenges that need to be overcome, or cultural challenges, and what kinds of support structures need to be put in place to make sure that these solutions are sustainable, that we wouldn't go back in a month's time and the solution will have fallen apart, or people would have abandoned it. So as you can see, it was a really multidisciplinary project in terms of the different objectives of the study. So in terms of the first objective, the first thing we did was we did a systematic review, and we looked at all the studies in terms of water treatment, and we found 67 studies going back to 1902, looking at the different forms of water treatment. And really there was quite a lot of information, quite a few studies on different ways that you can make water safe just by storing the water for a certain amount of time, for example, or heating it. But there was quite a lot of difference in terms of the way that some of these studies were conducted over the past 100+ years. And so one of the conclusions that we came from this review was that really there's a bit too much inconsistency in the experimental methods and insufficient data to be able to come out and actually say to someone in Tanzania or Ethiopia, here's the chlorine dose that you need to be assured that your water is safe, for example. So the first thing that we did is we went to the lab, both in Imperial and also with partners overseas. And we did some chlorination experiments. And so we asked questions like, how much chlorine do you need to inactivate cercariae? What chlorine concentration and contact time do you need? And also, what's the effect of some of the water conditions, like the water pH and the temperature of the water? And we did this thinking that even in the lowest income situations in the countries where we were partnering, there is good access to, for example, liquid bleach, which could be used as the source of chlorine. And so this graph here shows the relationship between the log inactivation of the cercariae. So 1 log means 90% kill, or 2 log means 99% kill. And on the x-axis is what's called the CT value, which is the product of the chlorine concentration and the contact time of the chlorine with the water. And on A and B here is 2 different temperatures that we tested of the water temperature. And so we also did some controlled experiments in the lab with infected snails that our partners from the Natural History Museum maintained and supplied to us. And we also did the same experiments, in this case in Tanzania, with Lake Victoria water, with real cercariae being shed by snails there. And we found very consistent relationships really between what we were finding in the lab and what our colleagues in Tanzania found with Lake Victoria water. And so we were then able to say, well, what is the CT value, the product of chlorine concentration and contact time, that you need to achieve, say, 2 log 99% kill of the cercariae? And it turns out that it's about 30 milligram minutes per liter. And so you can see in this table here how that compares against other waterborne pathogens. Most of these are drinking water pathogens, where there's been quite a lot of research into those previously. And really the take-home message is that cercariae seem to be slightly harder to kill with chlorine than those other waterborne pathogens, but it's still something that's practical and economical to do, using the types of chlorine that are available in low-income countries. And so we then, once we develop this sort of robust understanding of how much chlorine you need, the next thing is, how do you translate that into practice? And a lot of people in low-income settings, they buy their water from water vendors, like you see here on the bottom of the slide, a guy who might just go to a lakeside and collect water in jerry cans and then go around and sell those containers to people. And so our sort of scientific recommendation, it was 1 milligram per liter of chlorine for 30 minutes. But the practical challenge of that is that quite often there's no way to measure chlorine residual in these kind of settings. But we were able to think of some ways that you could have some standard sort of rules of thumb in terms of how much bleach, for example, you'd have to add to one of these jerry cans based on the concentration of the locally available bleach. So that was an interesting challenge also, to take sort of laboratory studies and then think about how would you then explain this to somebody who is providing the water in terms of getting it right, getting the dosing right, to ensure that enough chlorine is added, not too much, not too little. Another thing we studied was sand filtration experiments. So here the idea was, what design parameters would you need to prevent cercariae from passing through a simple sand filter, like the ones that people might already have access to for purifying their drinking water in their homes. So again, we did some rigorous experiments where in both the UK and with our African partners, to figure out what depth of sand do you need, what sizes of sand are effective to filter out the cercariae, and also what's the filtration rate, the rate at which you introduce the water to the filter, so that you get effective removal of the cercariae. And again, we were able to determine the sand sizes, the filtration rates, and the sand depths that are effective at removing cercariae, as shown in the table here. And basically, as you'd expect, if you go to larger sand grain sizes, you need to have a greater depth of filter in order to get effective removal. And then the other type of treatment that we looked at was UV disinfection. And there's a few different ways that UV might be able to help. One, it would be to use what's called SODIS, or solar disinfection, to fill up containers, plastic containers, for example, with water, and expose them to sunlight for enough time to purify the water. But we also thought about, nowadays there are fairly inexpensive small-scale UV disinfection units that you can buy. Some of them are even solar powered, so you don't even need a stable power supply. And potentially you could treat water, from a transmission site like a lake or a river, and pass it through one of these UV reactors to purify the water for washing or bathing purposes. So that was really interesting research also, and we found here that actually cercariae are probably quite a bit more resistant to UV than some of the waterborne pathogens for which we already have data. But sort of a caveat there is that it's actually very tricky to measure cercarian activation by UV light. One of the sort of, I guess, molecular techniques that was involved in this research was we did an assay to actually measure the damage to DNA of cercariae that was being caused by exposure to UV light. And that was a way to quantify the amount of damage that was being done to them, and be able to tell with a bit more certainty that when the cercariae were dead and were no longer infectious. Otherwise, it's sometimes hard to just look under a microscope and tell whether a cercariae is squirming around. Is it still infectious, or is it not something we need to worry about? So that was sort of the water treatment part of WISER, the sort of techie engineering-y part. And as I said, we also collaborated with Paul Fremont's group in the Department for Infectious Disease, who's a synthetic biologist. And his team also were part of this project, and we're looking at, again, developing some type of novel way to collect water samples and be able to tell whether there are living infectious cercariae in the water. And they looked at two different ways of doing this in terms of developing biosensors. One would be to look for certain types of enzymes that would be in the water that are associated with the cercariae, and the other would be to try to detect the specific DNA of the cercariae. And so one of the approaches they use is to develop what are called cell-free based biosensors, and it's basically a colorimetric output that occurs using the bits from inside a cell, let's say, which is going to produce, as I said, a colored output when it comes into contact with a target schistosome enzyme, which I believe is called elastase, which is the enzyme that the cercariae produces when it's going to bore into your skin. So that was one way to do it, was to try to measure this enzyme associated with the cercariae. And then another set of biosensors that they developed was to look at the specific sequences of the DNA of the cercariae in the water. And again, that produced a sort of colorimetric output by having a probe that you would attach onto the sequence. So that was really interesting in work, and actually showed that it could work in the lab setting. I think there's still a bit more work to be done to show that it can work in the field, and you know, whether it can provide quantitative measurements of cercariae concentrations or just sort of a presence or absence kind of test. But I think over the course of the project, at least, there was a lot of progress that was made, and I think it's actually a very exciting application of synthetic biology, not just for schistosomiasis detection, but also detection of other types of parasites in water that are otherwise difficult to detect. And then the last part of the WISER project, the last objective, was really, again, going back to what I said about what are the limitations of water and sanitation and hygiene. And even if we had those things implemented, there could still be a problem with infection in a community. And really, these engineering solutions will be ineffective unless you have community buy-in. So the community understands the risk and understands the role of water and sanitation, hygiene, and preventing the infection. And so one of the things, for example, that we saw in some of the case study communities was that people might already have access to safe water, actually, but they might still prefer to use the contaminated water source for social or cultural reasons. So, for example, there were some villages where an NGO, a non-governmental organization, had installed a lot of groundwater wells, which were completely protected. There was no risk of getting schistosomiasis from that water. But in some cases, people didn't like the taste of the water. Maybe it had a little bit of iron in it, for example. Or in other cases, you know, we found that sometimes the women, they prefer to go to the lakeside to wash clothes together in the morning because it's part of their daily routine. It's part of their social routine. You know, it's their opportunity to catch up, find out how the kids are doing, and moan about their husbands and that kind of thing. And you don't get that same experience if everybody's queuing up to fill buckets from a tap on a groundwater well. So it's really important, you know, again, no engineering solution is going to be effective unless you work with the end users in the community and understand, you know, what it is they want from the solution, and you know, how that's going to affect the effectiveness of the solution as a public health barrier. And so part of the WISER project also involved partnership with an organization called Acting for Health. And Acting for Health are all about using theater-based methods to communicate health messages or to raise awareness about certain health challenges. And it turns out that actually by using theater-based methods, you can actually understand a lot more about a community and their relevant water contact practices and their beliefs about the disease, which you wouldn't really get if you did a traditional sort of survey-based study, if you just went around and asked people. So one of the approaches here that was tested as part of the research was that colleagues from Acting for Health went into selected communities and worked with some of the community members to put together a play, which actually had some vignettes which expressed typical situations in their communities which were relevant to infection risk, and also address some of the beliefs that people had in the community, which might not have been completely correct about transmission of the disease. So for example, we found that in some communities, there was a belief that by taking the drug, that the deworming drug, that it could cause impotence in the men in the community. That's a belief that if you were designing a questionnaire, you wouldn't think of really asking that question. So they developed a whole play, a whole sort of short vignette to then perform to the rest of their community, to sort of explore that issue and to sort of communicate in their own way some of the flaws in that understanding. And we also videotaped these plays, and we then were able to show them in other communities and also study, from a social science perspective, whether it's more effective to do a live play with people in a community who or whether a video of people from a different community is equally effective in communicating these health messages. So that was really a pleasure for me to be a part of, also just to understand the role also of social sciences in helping us to raise awareness and to ultimately make the engineering and the science interventions more effective by raising that awareness. So that's just about all I wanted to say today, and to wrap up with a few take-home messages. And I think the first one is really a general one, which is just that scientists and engineers have important roles to play in tackling major global challenges, such as eliminating tropical diseases. And of course, we all need to be really good at our own disciplines, but I think really I'd encourage you to also think about interdisciplinary and also international collaborations, including with local affected people, not just researchers, but people who are actually affected by the problems. Because that's really where I think the really effective solutions and the more holistic approaches to solving some of these global challenges are going to arise. And as I mentioned earlier, it's certainly the way that a lot of international organizations are increasingly thinking about these challenges, is that it has to be an intersectoral, interdisciplinary approach. Also, I would say that scientific discoveries and emerging fields of science, for example, molecular science, but also synthetic biology, as I mentioned, I think there's huge potential to yield useful new tools which can contribute to solving some of these problems. So, for example, we saw the example of the potential for sensors for detecting parasites and really making our ability to assess the risk, the environmental risk, much easier by applying some of this advanced science. And also the fact that in terms of going back to schistosomiasis, really, as I said, the WHO now recommends that it's not good enough, or it's not going to be likely to lead to elimination of this disease if you're just applying mass drug administration to large populations. It's fantastic that the drugs are available in such large supply and that they're donated, but it's not really a sustainable solution in the long term. And so we also need environmental improvements and also health education and awareness raising, to really maximize the value of these drug donations and make sure that reinfection doesn't occur so rapidly. So those are some of the take-home messages from my presentation today. Some acknowledgements of some of the researchers who contributed slides, and of course the funders. And just a final slide also to mention that Imperial now has something called a Global Development Hub, which was launched by the Deputy Secretary General of the UN in April of last year. And we're sort of starting to get going now as we come out of the pandemic. And really, one of the key missions of the Global Development Hub is to try to encourage these kinds of interdisciplinary research and international research collaborations to address sustainable development goal focused research in the broadest sense. So not just health, but looking also at all of the sustainable development goals. So yeah, I'd encourage you also to check out the Global Development Hub. We run regular webinars like IMSE does, focused on particular SDGs, and I'd encourage you to also get involved in the hub. Right, thank you. That's it for me. Thanks, Nick.
Yeah, thank you, Michael. That was fantastic. I mean, very exciting presentation and so much information there. And let me see if we have any questions in the Q&A. I'm not sure if we do.
Okay.
But I have a question I want to ask.
Okay.
So I didn't understand the different social perspectives. Was it because people were assured that they can be treated by pharmaceuticals and that's why maybe they didn't care so much? Or was it that they didn't, what was it that drive the social practices that were not supportive of eliminating the disease?
Yeah, it sort of varied from community to community, and that was sort of one of the interesting outcomes too. But some communities, they just had completely unscientific understandings of how the disease was transmitted. So in some communities, there wasn't even a recognition of the link to water contact necessarily. But in other communities, as I said, even when that was made clear to people, sometimes for social reasons, people continue to have contact. And I think that is partly down to maybe failure of the engineering solution to take into account the preferences of people. Maybe they didn't like the taste of the water or didn't understand the social dynamics. And I don't think in many cases it was sort of a case where people thought, oh, I can just take the drug and be treated. I think people in most communities, it was a case of raising awareness about the disease and then also making sure that they had access to the drug and uptake of the drugs would then increase in these communities alongside improving the water and sanitation. But yeah, it was complex. It varied from community to community, really, I would say.
That's great, thank you. There is a question now, and it's where next for this project? What's the next challenge?
Yeah, so one of the things we're working on now is the link between hygiene and schistosomiasis. So for example, if you're washing your clothes with soap, with different types of soap, does that protect you from the cercariae? And there is some sort of anecdotal evidence that it does provide protection. But again, there isn't really sort of robust scientific evidence to suggest that that's a good strategy for preventing the disease. So we still have some active research now, which is looking at different types of soaps that are used both for laundry, but also for bathing, and how protective those are against cercariae, so that hopefully we can influence policy to sort of say, if you're going to implement hygiene in these communities and give out, bar soap or different types of soap, here's maybe some of the messaging that has to go along with that to maximize the effectiveness. So that's something we're involved with at Imperial, but there's a lot of research on each of the different aspects of prevention of the disease that's still going on globally. How to effectively deliver the drugs to big communities and not miss certain people, or what are the most effective forms of toilets, of sanitation to prevent the disease, or are there safer types of molluscicides to kill the snails than the ones that are commonly used currently? So a lot of research still to do.
Yeah. And when you boil the water, do you kill them?
Boiling the water does work. Yep. Of course, that relies on people having access to fuel, and then also you have then hot water, which you have to wait to cool down, and also that might be good for maybe producing water for maybe small volumes of water. But if you're talking about bathing or doing laundry, you'd need quite a large volume of water to be boiled for that. So
because it's just contact, you don't have to drink it, right?
It's contact mainly. Yeah, drinking isn't so much a route. Yeah.
Okay. Well, I don't think we have any more questions, and thank you so much for such an informative presentation. A recording of this webinar will be put up on the YouTube channel of the Institute. You can find a link for in the chat, hopefully. I'm going to put up there. But again, thank you, Michael, and well done. Thanks.
Thank you.